Again, today is Unit 5, Day 5. So you'll scroll down once you're in modules to Unit 5. Today is October 12th. 6 minutes. Good morning, Diana. Okay, so we're about halfway through the warm-up. It's a short one, only 5 questions, so this should be ample time to complete it. So go ahead and start wrapping up. 3 more minutes. seconds. So go ahead and submit your warm-up and start heading back to the Google Meets where you'll be able to see my screen. Great. So welcome to another week, folks. It is Monday, October 12th, 2020. So we're moving through this quarter, is almost at an end. We'll be heading into the second half of our class later this week. So time is flying. As you all know, I like to start off each week with a brief message of positivity. And based on where we are in the month, with the end of National Hispanic Heritage Month coming on Thursday, and with the Supreme Court of the United States in the news a lot today, especially as we are potentially preparing to nominate another Supreme Court justice, I did want to talk about this person specifically. Does anyone have any idea who this is? Huge brownie points if you know. And thank you for joining us, Stephanie and Alicia. She looks familiar, Gabby says. Okay, that's good. I'm at least happy to know that you've seen this face.
I've seen that lady today on the news heading of something, but I don't know exactly who she is.
Good. So this is Puerto Rican Supreme Court Justice Sonia Sotomayor. And so the week that I want to choose, the quote that I want to choose this week is from Justice Sotomayor. Just a little bit about her, a little background. She earned her bachelor's degree from Princeton, very prestigious school. She earned her JD, which is a law degree, from Yale, again, very prestigious school. She was appointed to a lifetime position on the Supreme Court by the 44th president, President Barack Obama, in 2009. And so the Supreme Court actually is a lifetime position, meaning that the only way that any of the Supreme Court justices will lose their position is if they retire, if they die, or if they are impeached, which has never happened in the history of the Supreme Court. So that was a big deal. And it was also a big deal because she was the first Latina and only the third woman in the history of our country confirmed to the Supreme Court. So the quote that I've chosen from her says that, "I have never had to face anything that could overwhelm the native optimism and stubborn perseverance I was blessed with." Okay, so what she's saying is through all the trials and tribulations she's had to face in her time as a student, in her time as a district attorney, in her time as a Supreme Court justice, the fact that she was born with this sense of optimism, this glass half full attitude, as well as a perseverance, meaning she was willing to dedicate herself to hard work. Those things combined meant that there were no trials or tribulations that would ever conquer her. All right. And so even though some of us don't have sunny dispositions, we don't have that glass half full mentality, I do think that we can start to work hard for the things that we want, or continue to work hard for the things that we want. And as we do that, as we start to rack up some successes, as we feel good about ourselves, our sense of optimism will increase. All right. And then you'll start to notice that you approach any challenge, no matter how difficult it is, with this sense that you can overcome it, that you can persevere over it. All right. And so that's the quote that I want you all to start this week with. Even though you might feel as though at this point in this semester, as the first quarter wraps up, you're not exactly where you want to be, I want you to take some time at the beginning of this week to think about the things that have gone well this semester. Think about, in this time of pandemic, in this time of virtual learning, all the ways that you've adapted to meet this challenge. And think about how through perseverance, you've gotten to this point. And I think with that mentality, with that optimism, you'll head into the second quarter ready to head into midterms. Let's take care of midterms first, but head into midterms and then head into the second quarter with the mentality that you'll need to do even better. And then one more thing before we jump into our actual biology lesson. Today, as many of you may know, is a holiday. Does anyone know what holiday today is, even though we're still in school?
I'm sorry, but no.
Okay, no problem there. Maybe that's a good thing based on what the holiday is, depending on what your perspective is about the holiday. But
Columbus Day?
historically, what'd you say? What was that, Castro?
Columbus Day?
You were 100% correct. So historically, we have celebrated Columbus Day today. In fact, it was really recognized on Friday, but some people have today off in celebration of Columbus Day, which of course recognizes Christopher Columbus. It also has historically recognized his quote unquote discovery of the quote unquote new world. But the reason I bring this up is because I want you all to be aware of the fact that for the last 29 years, various cities around the country have used today to celebrate Indigenous Peoples Day instead. Okay, and Indigenous peoples are a group of people who we have talked about in the past and who we will continue to talk about as their unique struggles and as their unique abuses have been probably overlooked in the history of our country. But so a lot of people over the last 29 years and even before that across the country, starting in California, have chosen to recognize this day as Indigenous Peoples Day, to celebrate the victories of Native American people, to celebrate the Native American culture, to celebrate all the ways that Native American people have impacted our contemporary culture. And so this I won't say started, but it really came onto the national agenda when in 1977, attendees at an international conference decided, quote, to observe October 12th, the day of so-called discovery of America, as an international day of solidarity with the indigenous peoples of the Americas. So Columbus, of course, in 1492, landed in what he thought was India, as he was in search of, thank you, Angela, I appreciate you being here. He landed in what he thought was India as he was in search of a route from Europe to India. Of course, there was a spice trade that was taking place, and it was extremely economically beneficial for Europeans to have some trade route, some way of getting the spices from India to Europe without having to go over vast amounts of land. And so he thought he had arrived in India, but in reality, he had actually landed somewhere in the Caribbean. Today, we believe that he landed somewhere on the coast of Jamaica. And from that point forward, we started a history of colonization that was extremely, extremely damaging to Native American populations, eventually killing off about 90% of the Native Americans, not only in North America, not only in the United States, but also South America as well. And so it's important that we acknowledge both sides of the history. Yes, Christopher Columbus did land on the Americas, and he opened up huge potential to create what we now know as the United States of America and many other countries. But we also have to acknowledge that indigenous people, the people who were here first, the people who didn't discover America, but the people who had been here for many thousands of years, they also have a history that needs to be acknowledged. And so that's what I'm thinking about on this Columbus Day. That's what I'm thinking about on this Indigenous Peoples Day. And that's what I want you all to think about as well. That being said, let's go ahead and take out our notebooks. Let's write down the title of today's lesson, which is cell specialization. It's kind of a difficult word to say. Let's also write down today's date, October 12th, 2020. And because we want to take efficient and effective notes that are organized in a way that will allow us to come back to them at some point, let's make sure we write down that it is Unit 5, Day 5. And as you all are writing, I'm going to go ahead and read the objectives and, well, there's only one objective and the essential question. So our objective is biologists will be able to explain how instructions in DNA lead to cell differentiation, which means that specialized cells perform specific functions. That's what we're going to learn about today. And then the question that we will seek to answer by the end of the lesson: how do the cells in multicellular organisms perform so many diverse functions? So today we're thinking specifically about multicellular organisms, meaning we're not thinking about bacteria so much today. We're thinking about organisms that are made up of many different types of cells, like humans. All right. So quick review here. On, I believe, Wednesday or Thursday of last week, we talked about transcription. And transcription is the first step in this process of protein synthesis, the process of making proteins. And this is so important that we call it the central dogma of biology, meaning it is the most important principle, as Castro told us. That's what dogma means. It's a principle, the most important principle in the study of life. And it states that genes code for the proteins we make, meaning they are the instructions. They are the instruction manual to make proteins. The first step of that is transcription. Taking the DNA that is in all of our cells and transcribing it to a strand of mRNA, messenger RNA. And then we know that the second step is translation. Taking a strand of mRNA and translating it from a language of nitrogen bases to a language of amino acids. That's why we call it translating. We're putting it in a different language. The first step, same language. DNA to RNA, they're both nucleic acids. Second step, two different languages. From a nucleic acid with nitrogen bases to amino acids. Again, the goal of this is protein synthesis. We have to be thinking about this process as trying to make proteins. That's what a cell is ultimately trying to do. That's what all cells are ultimately trying to do, make proteins. So we should be clear on that. We've reviewed with that before. If we have time at the end of the lesson during the synchronous portion, then we will come back and play this Kahoot. But let's go ahead and knock out the notes first. So again, the portions that are highlighted in yellow or in green are the portions that you should write down depending on your preferred language. So here we've got a definition for gene. Would anyone like to read that definition for us, please?
I'll read it.
Thank you.
A small section of DNA that codes for a specific protein.
Excellent. And so as you all write down that definition, I just want to read a fun fact to you. So in human DNA, the DNA that we all have in all of our cells, there are approximately 30,000 genes. And each gene makes an average of 3 proteins. All right, so that's a lot of different proteins. Of course, we really only focused on 5 so far. We talked about hormones, insulin, antibodies, enzymes, and hemoglobin. But there are so many others that do things that maybe you don't even have to think about, you've never thought about before. And those proteins are produced by genes. They're coded for by genes. So even though we have this huge, long DNA in our cells, it's really, we should be thinking of it as being divided into many, many, about 30,000 smaller sections that actually code for proteins. Okay. Any questions based on this slide?
No.
Thank you, Ayana. All right. So the next most important definition that we have in this lesson is the definition of gene regulation. Would someone else, other than Ayana, even though you did such a great job on the last slide, can someone else please read this sentence, this top sentence? Angel, I haven't heard your voice in some time. Omari, I haven't heard yours in some time. Dante, we have perfect attendance today. I want to congratulate you all for that.
Gene regulation is the process of activating and deactivating certain genes.
Thank you so much, Joseph. So we want to think about gene regulation like a light switch. So our DNA, human DNA, we have 30,000 genes in our DNA. And at any given time and in any given cell, only a small portion of those genes are actually activated. They're actually turned on. So if you thought about it, if you had a house that had 30,000 rooms in it, you would probably only confine yourself to a small space. You might use 4 of those rooms, the bathroom, maybe 2 bedrooms, maybe 1 bedroom, a kitchen, and a living room where you have your TV, right? So at any given time, you have those 4 rooms turned on and everything else is turned off. You don't want to waste the energy. Our cells are the same way. They're not going to waste energy turning on genes that would produce proteins that they simply don't need. So if we think about this as a light switch, our genes and our cells can literally turn some on while most of them remain off. And in some cases, there are some genes that are naturally on that need to be turned off. So we can go in both directions. Some are naturally off and can be turned on, meaning they can be activated. Some are naturally on and can be turned off, meaning they can be deactivated. All right. But we must keep in mind, this is so important. All cells have an entire copy of DNA. So it's not that some cells have this portion of DNA, some cells have that portion of DNA, some cells have the whole thing. All of the cells within an organism have the entire copy of DNA. It's just that some of them have some portions of that DNA turned on and other portions turned off. And when I say portions, what I'm talking about is genes. So this is why gene regulation is so important, because we don't have half of the DNA over here, half over there, a quarter over there, a third over here. We have the whole thing. We have the entire copy. We just turn on and turn off some genes along the copy. So gene regulation controls what proteins are produced in each cell, because each type of cell is ultimately responsible for producing different proteins. So as you all finish writing that highlighted portion, I want you to take a look at the image that you have in the middle of this slide. We can see that there's a long strand of DNA here. And along this strand of DNA, I've got 1, 2, 3, 4 genes. We can see that each gene is only a small portion, a small segment of the DNA. 2 of the 4 genes are turned on, and we can see that they're producing 2 different proteins here. Who knows what exactly they are, but they're producing 2 different proteins. The genes that are turned off are not producing anything. Okay, so again, we've got to think about this literally like turning on a light, turning off a light. If we can, we will conserve the energy by having the genes that we don't need off. If I don't need to produce a protein, then I don't need the gene that codes for that protein to be on. I'm just going to keep it off. Are there any questions about this gene regulation process before we switch into the next concept?
So those are all different genes, or are those the same genes on and off?
These are 2, based on the shape. Remember, structure determines function, and this is something we'll talk about later in this lesson. Based on the shape of these proteins, we can assume that they are different and do different things. Now, it is the case that sometimes, and this is a good question, Ayana, because this will open us up to the next concept. But it is the case that sometimes there are genes that produce the same protein. So there might be multiple genes on 2 different chromosomes or on 3 different chromosomes that all produce the same protein. Can anybody think of why that might be? Why would I want to have the same protein being coded in multiple places? Any idea why do our cells have multiple genes that do the same thing essentially? Gabby, I see you putting your thinking face on. Okay, so that's something that we should think about. I don't want to give you all the answer right now because I know you're capable of thinking about that. In fact, we talked about it a little bit last week. But as we go into tomorrow's lesson about mutations, you all should be able to answer that question. So let's keep that in mind. Why do our cells need to produce the same protein in multiple genes? We'll think about that. All right. Gene regulation is controlled by signals. So cells are constantly communicating with each other. And the way that they communicate is via these electrical signals and also via chemical signals. So neurons in our body, whether they be in our brain, in our spinal cord, or all the way in our fingertips or in the tips of our toes, are responsible for carrying electrical signals that allow our body to communicate. Our cells.
Sorry.
Go ahead.
Do neurons allow us to feel pain as well?
Yes, yeah. And if this is something you're interested in, I would definitely encourage taking the anatomy class or taking the AP biology class where we talk a little bit more about this and in greater detail. But Ayana, you're 100% right. Neurons are what allow us to feel things. We've got what's called a motor nervous system, which allows our body to interpret a number of sensations: pain, heat, pressure. And then we've talked about hormones as well. Hormones are the chemical signals that our cells use to communicate.
Wait, can you repeat the question again?
Oh, there was no question in that case.
Oh, did you know you could be?
Go ahead.
Did you know you can be born with this? I forgot what it's called, but it's where your nervous system, you don't feel any pain at all.
Yes. And in fact, not only can you be born that way, but it can also be the result of injury. People who have experienced paralysis as a result of injury or as a result of some type of disease, they lose their motor and sensory functions. So I think I said it backwards a couple of minutes ago. Motor is what allows us to move things, to move different parts of our body. Sensory neurons allow us to process information, to feel things. So if there's some type of damage, depending on where the damage took place, you might lose a small amount of sensory ability, or you could lose sensory ability throughout your entire body. So the central nervous system and the peripheral nervous system, again, if this is something you're interested in, neurology, or just anatomy in general, then I would definitely encourage you to take that anatomy class or AP biology. Thank you for the question, Ayana. All right. So some of you might be wondering why we put such an emphasis on genes, because perhaps you've heard that a person's environment, like their diet or the activities that they engage in, or the amount of chemical or radiation exposure that they have, can also affect who they become and the diseases that they develop. And that's true. There is always a constant interplay between our genes and our environment. This is something we'll talk much more about in the 6th unit of our class. But both our genes and our environment affect the proteins that our cells produce. They're both really important. Again, we've got to be thinking about all of these processes as affecting the proteins that are produced. That's what a cell is ultimately trying to do. All cells are trying to produce some specific type of proteins. So for those of you who are interested in and considering potentially becoming scientists and studying these things, what you'll see is that many scientists do research on twins, because twins are about as genetically similar as you can get, especially identical twins. Their DNA is 99.999% alike. And so we can study them knowing that their genes are almost exactly the same, but their environments might be slightly different, especially as they get older. And one twin goes to college on the East Coast and the other goes to college on the West Coast. Or one twin plays basketball, whereas another twin is an esports fan and plays video games. You know, two different skill sets required. So we can start to see how diet, activity, and exposure to chemicals and radiation affects the genes. For example, in this image, we've got two identical twins, both born with the exact same gene that absorbs fat twice as fast as the average person. And this means that they both are more likely than the average person to become obese. But the twin who chooses to eat healthy foods that are low in fat does not actually become obese. She doesn't give her body the opportunity to absorb that fat. The other twin chooses a different diet, and that gene does have an impact on that twin's body shape. All right, so there's a constant interplay between our genes and our environment. Not only do our environments impact our genes and how our genes are able to produce proteins, but who we are genetically also impacts how we engage with our environment. So both of these things are in an interplay. And this is a good segue to talk about Breast Cancer Awareness Month, which is October, the month we are currently in. And I want someone to unmute themselves and answer this question. How might a person's likelihood of developing breast cancer be affected by both their genes and their environment? Does anyone have any thoughts about that? How might a person's likelihood of developing breast cancer be affected by both their genes and their environment?
Well, I think that if you already have people in your family that have had breast cancer before, you are more likely to get it. And based on your environment, I guess, I don't know, if you get exposed to like lots of UV and radiation waves and different stuff that is harmful to you, it can also cause breast cancer to appear.
Wonderful, wonderful answer. That is 100% correct, and that really summarizes what I was getting ready to talk about. I'll still talk about it though, but that was great, Ayana. Thank you. Does anyone else want to add to that? Okay. So genetically, and you all do not need to write this down, but this is just good to know as it relates to Breast Cancer Awareness Month. Genetically, there are two genes located on two different chromosomes that have been shown to impact a person's likelihood of developing breast cancer. So on the 17th chromosome and on the 13th chromosome, we've got these BRCA genes, BRCA. And BRCA, of course, stands for breast cancer genes. So everybody has these two BRCA genes. But people who have mutations in these genes are shown to have a much increased likelihood of developing breast cancer. So if we look over here, the BRCA gene 1 on chromosome 17, if a person has a mutation on that gene, their chances of developing breast cancer are as great as 65% by the time they're 70 years old. So that's high. That's very, very high. The BRCA2 gene, which appears on the 13th chromosome, a person's chance is 45%, whereas people who have neither a mutation on the BRCA1 nor a mutation on BRCA2 only have a 12% chance of developing breast cancer by age 70. So here we're seeing the genetic impact, and some women have actually undergone genetic testing to determine whether or not they have these mutations. And once they know that, they can make a preemptive decision about what they want to do to potentially treat their breast cancer. Some women have actually gotten proactive mastectomies, which means that they surgically remove their breast tissue so that they cannot develop the breast cancer. And they do that because they know that they have a mutation on their BRCA genes, and it's very likely that they will develop breast cancer. Probably the most famous example of a woman doing that is Angelina Jolie. But in addition to those genetic causes, just like Ayana said, there can be other causes that increase a person's risk of developing breast cancer as well. So late childbearing.
Can I add something too?
Go ahead.
I was about to say what you intake, but I meant like not just like alcohol and drugs, like food as well. Some food can affect you.
Yeah. So this is just some of the causes, but Ayana is right that diet can also impact a person's chances of developing not only breast cancer but any number of cancers as well. But these are the specific causes for the top specific causes for breast cancer. Late childbearing, so women who have children after the age of 35 are at an increased risk of developing breast cancer. Doesn't mean that it's certain to happen. It just means that the chances are greater. Women who start their menstrual cycles at an earlier age or who go through menopause at a later age are also at a greater risk. Okay, so we see that there's this interplay again between genes and environment. Both of them work together. They collaborate to ensure that a person's destiny is not totally determined by either one. All right. Now, I do want you all to write this down, and I apologize that I did not translate this slide. Would anyone like to read this definition, please, for us? Someone, anyone? I see Gabby's writing. Maybe Gabby, you can read it when you're done writing for us. Someone whose mic is working. Kiki, we haven't heard from you. Hopefully you're having a good Monday. Can you read the definition for us? Or Jalen?
I can.
Thank you, Stephanie.
The process by which cells produce specific proteins and therefore develop different structures, which result in different jobs and abilities.
Excellent. That was read so eloquently and clearly. I appreciate you. So, the process by which cells produce specific proteins, again, that is the number one most important function of any cell, to produce some type of protein. But cells produce many, many different types of proteins. And so the proteins that they produce determine how they're shaped, and then also determine what job they can do, what abilities they have. So this is why structure determining function is something that I talk about all the time. A shape the shape of a cell is ultimately going to determine what job that cell is able to do. And the shape of the cell is determined by the proteins that the cell makes. Let's see, is Gabby still writing? Nope, looks like Gabby's done. So I'm gonna move on in 3, 2, 1. All right. So maybe you all have heard of stem cells. In the last 20 years or so, there's been a great amount of scientific research about stem cells because they have huge potential. And the reason that they have huge potential is because they are undifferentiated. So we just talked about what it means for a cell to be differentiated. That means that it produces a specific type of protein, which gives it a specific job. But these stem cells are not yet producing any specific type of protein. They have the ability to develop into any number of different specialized cells. And so these stem cells are important because let's say that a person is born without the ability to produce insulin. And that means that they have type 1 diabetes. We can use stem cells to step in and serve in that job for that person because they were born without the ability, their kidneys weren't given the ability to make insulin. So we can use stem cells, develop those stem cells so that they become the exact type of cell that produces insulin. And then we can allow that person to start making their own insulin rather than having to spend thousands of dollars a year buying the insulin. All right, so scientists around the world are in the process of researching what exactly these stem cells are capable of doing. In fact, in some cases, people who have become paralyzed are relying on these stem cells because when you lose nerve cells, our bodies don't have any way of rejuvenating those. So if you have cells in your brain or in your spinal cord that are damaged, our bodies cannot replace those by themselves. But research is showing that stem cells do have the potential to serve as a replacement. So this is a big deal. And it's a big deal because these stem cells are undifferentiated. Not much to write there. But my question is, this is taking it back to Unit 3. What process are we seeing in this image? If we're moving from left to right, and then we're moving down here, what process are we seeing here?
Is that mitosis?
Beautiful. Yes, that is indeed mitosis. How could you tell, Ayana?
I looked at each and every one and I saw that they were splitting up. And the first thing that came into my mind was mitosis.
Good. So we can see that the cells are splitting up, as she says. They're reproducing. We started off with 1 cell, then we ended up with 2, then we ended up with 4, then we ended up with 8. So this is classic cell reproduction, and in this case, this is mitosis. Good job. All right, so cells start out as undifferentiated, but as their genes, we're going to take it back to gene regulation, as their genes are regulated, that is how they become specialized. And that is how they develop diverse functions. So your cells start out as stem cells. And then as they decide which proteins they're going to make, as they turn on some genes and turn the other genes off, they start to change shape, they start to develop specific functions. And then that's what allows them to do specific jobs for our bodies. So a stem cell might develop into a muscle cell. It might develop into a fat cell. Oops, I'm sorry. Might develop into a nervous cell, an immune cell. The way that our cells are able to determine what job they're going to do is by relying on that communication, whether it's electrical or chemical. So your cells are talking to each other. And we talked a little bit about this, but in the development of an embryo, when we start off just as a clump of cells, the cells are starting off as stem cells, but they communicate with each other. They say, okay, well, that group of cells is located kind of at the top of the body where we want the head to be. So they need to start developing into nervous cells, brain cells, and some of them need to become cells for eyes. And eventually some of them become taste buds. And eventually some of them become eardrum cells. But those cells over there are located near the back of what we want the body to be. So they need to become spinal cord cells. All right, so the cells are talking to each other. They're sending hormones to one another and saying, based on where they're located, you need to become this, you need to become that, you need to become this. So that over the course of about 40 weeks of gestation, 40 weeks of development, which starts off as a clump of cells, and none of those cells has any specific job, ends up as a human being that has many, many different types of cells, each having its own job. Okay, so this only goes up to 46 days. In reality, it would be closer to 280 days. But we can see how early on in the process, cell differentiation, cell specialization is allowing the body to become what we can recognize as a human being.
I'm sorry to say this, but in the early days, it kind of reminds me of a fish, like a little tadpole thingy.
I'm so glad that you said that. That is going to come up in a later unit of the class, because if you look at many different species, when they're at this stage in their development, you know, they're only a few weeks in, maybe a month or so in, you really can't distinguish between the different species. So a human being looks like a tadpole, which looks like a turtle, which looks like a gorilla, right? So the reason for this is because all of these species rely on the same process of cell specialization. We all start off as just a clump of undifferentiated cells. And then as we develop, we all have, because we're all mammals, we all need spinal cords, we all need brains, we all need a heart. As you can see, this dense cluster of cells here is becoming a heart. So in the earliest stages, we just need to kind of check off the most important features: brain, spinal cord, heart, and then we start taking care of the other things that make us what we eventually become. Okay. Now, as we wrap up the lesson, I want to revisit the essential question. The essential question today was, how do the cells in multicellular organisms perform so many diverse functions? Can anybody unmute themselves and answer that question for me? Just give me a quick summary, 1 or 2 sentences. Maybe someone who I haven't heard from today. I know Gabby's mic isn't working, but surely it's not the case that 10 of you have broken mics. Maybe Jalen, maybe Dante, Olivia. How do the cells in multicellular organisms perform so many diverse functions?
They work together.
Say that again, Castro.
They work together.
And what do you mean by work together?
So they give like instructions, like you go like try to do the tissues or like the skin.
Okay, so they're providing instructions to one another, so they're communicating.
Yes.
How else? How does a cell know that, or how, once a cell gets its instructions, how does it choose to make a certain type of protein versus another one? How does it know how to do that? Okay, DNA is a part of it. And can we say a little bit more, Gabby, or anyone else? Because as we know, all cells have the entire copy of DNA.
It's because of their shape. Different shapes lead to different actions taken.
Excellent. So different shapes lead to different functions or different actions taken. And how do they end up? Again, we want to think about this as being a process of protein synthesis, making proteins. So how do some cells make certain proteins that give them a certain shape, and other cells make other proteins that give them a totally different shape? How do they do that?
Is it because of the DNA or what they're made out of?
The DNA is a part of it, yes, but let's think back. Let's think about, excuse me, what we were talking about earlier in the lesson, because every cell has the exact same DNA, but what is different about them? What's different about how they use their DNA? Think about my light switch analogy. You can turn it on and turn it off. What is that process called?
Is it because they have many different genes?
Thank you. So I'm glad you said that at the same time that Gabby sent that message. So many different genes. And of course, in human DNA, we've got about 30,000 genes. That number can vary across species, but we've got many, many different genes, as Ayana said. That means that in many different combinations, I can turn on some, turn off others. And that gives me a unique set of proteins. So my blood cells have a protein called hemoglobin. So they need to have the genes turned on to produce hemoglobin. But the genes that produce insulin, my red blood cells don't really need those genes turned on. They don't need insulin. All right, so this process of gene regulation leads to cell specialization, which is what Gabby typed to the chat. So I think that the exit ticket today, as well as the asynchronous assignment, will better help, well, I guess, instill these ideas in your mind. So the exit ticket, of course, is located just under the warm-up for the day and the Unit 5 module. And the assignment, the asynchronous assignment, is called Cell Specialization Practice. So I will be here on mute. You all have 17 minutes. Both of them are short. The exit ticket is 5 questions. The cell specialization practice is 3 questions that might be a bit more difficult, but certainly you should be able to get it done in 17 minutes. I will be here on mute if you need anything, and have at it. Joseph, do you have a question? I see you're off mute. Okay, folks, it is 11:35. So at this point, you should go ahead and enjoy your 30 minutes of lunch. And I hope you have a good rest of your Monday. I will talk to you all tomorrow.
So we'll be taking our midterm tomorrow.
Yes, we are scheduled to take